Positive results for everything, and you'll get to look at tubes like this for the exam. They'll have more solution in them than this because I want you guys to be sure and see the color. But the protein was tested with Bradford reagent, and it was bright blue. And you know, it was kind of like that burgundy purplish color when it didn't change, or like a darker purplish blue. But it turns bright blue when there's protein. And then this, it turned almost black. That was the starch being tested for. Yes. Yeah, that's Mom. You better answer. Hurry up. Us moms worry when the children don't answer. So this was testing for starch with the iodine. So remember iodine, and when it doesn't test positive with the iodine in it, it's like a pale yellow amber color, but it turns black if there's starch in it. And then you had the white precipitate with the barium chloride and the silver sulfate, or no, it was silver chloride and barium sulfate. I'll get it right in a minute. But yeah, so you had the white precipitate with each of those. And in fact, those are the negative reactions for the other two. Those, they tested positive in both because they went through the membrane. It's just there wasn't quite as much precipitate on that end as there was the other. But yeah, you see it's kind of like that purplish red color for the Bradford. And the iodine was like yellow, dirty yellow, so. But yeah, so you'll need to know that. And then also with the enzyme reaction, you had the glycol, it turned the precipitate brown. You need to know that the darker brown one, there's more enzyme action, the darker it is. 2, it's got a higher absorbance, the darker it is. 3, it's got a lower transmission, the darker it is. So I don't know if I can draw that to make sense either. So when we did the glycol, okay, and then on the other end of the spectrum, the lighter color is the inverse. So the lighter the color, lower activity. So with the enzyme activity, your darker color means it's higher enzyme activity and higher absorbance rate and lower transmission values. And then the lighter the color is, it's lower enzyme activity, lower absorbance, and higher transmission. So, and then if you're asked which the molecules diffuse the fastest through that membrane when you did dialysis, I'm bouncing back and forth, sorry. So when you were asked which one of these diffuse fastest, it's always the smaller. So and then the size of the membrane. Is the size, I don't know right in English today, of the largest molecule that passed through. So in this case, the largest molecule that passed through it was your sulfate, which was 96 Daltons. And this was 35. These were 40,000. 70,000. So when you're asked the size of the holes in the membrane, it's going to be the size of the largest that passed through, which was your sulfate ion. So you would say it's 96 Daltons. And the smallest diffuses fastest, so the smallest is chloride. Yeah, I printed this quiz to make it more linear, and I'm all over the place still, aren't I? I'm so hot and flustered. So the substrate for the enzyme peroxidase, we're back on enzymes again. Our enzyme was peroxidase that we used. They'll probably ask me that again. And its substrate, and this is the enzyme. Which is hydrogen peroxide. In this reaction, one of them got oxidized and the other got reduced. The enzyme, we had the glycol, which was our reporter basically. You could consider it, I don't think you'd really consider it a substrate, it was just the reporter to make the substrate be reduced. So the glycol is already in its reduced form going into the reaction, and it gets oxidized, and the peroxide got reduced into water. And then we use the spectrophotometer to measure absorbance. You're always measuring absorbance. I think transmission, they only use it if they're like doing wavelength light studies and you want to measure light transmission. But in this lab, we've not been measuring light. We've been measuring the absorbance of different molecules. So it's always absorbance. And then why do we use the germinating peas rather than full growth plants? It's because full-grown plants have chlorophyll already, like the Elodea leaves, and they're performing photosynthesis as well for energy. So we wouldn't get a clear view of how much cellular respiration they're doing because they'd be using as much carbon dioxide for photosynthesis as they are oxygen for respiration. Basically, it's because they perform photosynthesis in the germinating bees don't. Write out the balanced chemical equation that shows the reaction between water and CO2. How many of you had that memorized already? Did any of you have it and get it wrote down? All right. So the balanced chemical equation, and we'll try to do this without erasing stuff. So H2O plus carbon dioxide, which is what they were respiring. And I don't know if those were arrows going both ways or not. I guess I could look there. No, they're just that. And that gets converted to carbonic acid. Let's see about H first. H2CO3. And that, in turn, it's a highly polar molecule, so it dissociates hydrogen ions and CO3 ions. That's your reaction. And it's really intuitive because it's the exact number of these atoms, right? You're adding your H2O to CO2, and it gives you H2CO3, and that, because it's more polar molecule than it is, what's the other kind of bond that's more permanent? You guys remember your chemistry? This is more of a polar bond though. And that means it dissociates your hydrogen ions, and that shows for every molecule of carbon dioxide, you're producing more hydrogen ions, which is why we use the phenolphthalein to measure the solution, because that's a question there too, because it's a reporter for the pH of the solution. We started with a solution that was basic, about a pH of 9, 9.5, maybe higher, because I think you guys, if you had to do like 177 drops to clear the solution, it was, I think the pH was 10.6 on those. I apologize. So that's the information on that. So in the second one, what's missing from the formula for cellular respiration? What do cells need along with oxygen to produce that energy? What's it come?
It's glucose.
Glucose. That's it. So the missing thing in that reaction was glucose. And its chemical formula is C6H12O6. So that's what's missing from that formula. But be familiar with that formula and know all the parts of it. I thought that was a pretty good question. So yeah, the purpose of the phenolphthalein was to visualize the reduction of the solution pH. Because it's pink when it's a higher pH, so it's more basic, and it turns clear as it gets more acidic. And once it got clear, it's not acidic, it's neutralized. It's to the neutral pH of 7 when it gets clear. That's why we're able to just pour it down the sink. All right, here's the next one. This is the fun one. I've added all the steps in there for standardizing the spectrophotometer. So the first thing you want to do is set your wavelength for what wavelength you're going to use. And you guys remember what part that is on the spectrophotometer, right? The wavelength is that dial on top of the spectrophotometer. So the first thing you'll do is when you're going to measure something, you need to know what wavelength it is. You set it to the wavelength you want. That's of course assuming that you've got it turned on. You can plug that in. Yeah, because these need to be warmed up with someone's going to use them. Okay. All right. So set your wavelength. That's first. Then you're going to press your mode button. Nope, wrong one. Mode button and set it to transmission. And this, oh, it's got to be warmed up. Do they have to be warmed up to listen?
We don't have to wait.
Oh, there it goes. Yeah. You have to wait a second for it to like boot up, right? All right. You push your mode to transmission. Then you wipe your cuvette containing the Blake solution. Can you hand me a wipe? Here's my blank. I'm going to wipe the cuvette and place it in there. You always wipe the cuvette before you measure or set a blank, because fingerprints or dust will alter the readings. Then you press your 0A 100% T to set your blank. And what it does, that sets the blank to 100% transmission or 0 for absorbance. That's, so when you set the blank, it's setting the transmission to 100% and the absorbance to 0. That's that next question, number 7 down there. When it's standardized, the absorbance is set at 0 and the transmission is set at 100%. All right, so now we've set our blank. We're at 100% transmission. Now I'm going to hit mode and move it to absorbance, because that's what I'm going to be measuring. Then I take out my blank, put it over here, and take my sample that I'm wanting to measure and wipe it, put it in, and now I can take my readings. I covered all the steps, didn't I? Yeah.
Yes.
So did you write those in order? Be sure you have those clear and in order, because this right here, what I just did, that's part of what you'll get graded on the exam. You're gonna have to set the wavelength, set the blank, make sure it's changed to absorbance, and then measure your sample. Now in my lab Since we don't ever measure anything in transmittance, I will also accept if you skip the transmittance step and just set your blank on absorbance already, that's okay too. So you can leave out the whole transmittance step because everything we do is in absorbance anyway. So it's perfectly fine because you can set your blank on absorbance or transmittance because when you set it, it's always 0% absorbance, 100% transmittance, and they directly correlate on this machine. But it is a computer because I had students that measured in the wrong thing. They measured in transmittance. And in order to convert to absorbance, you got to remember that transmittance is a percentage. So you have to take the negative log base 10 of the percentage as it is a percentage. Like if they measured 85%, they'd have to take the negative log base 10 of 0.85 because it's 85%, and that would give them their absorbance value. And it's the negative log base 10 because, of course, it's less than 1. All right. Which one?
It's the
Number 12?
The net number of drops one.
Okay, the net number of drops. The net number of drops for each set of peas was the equivalent of CO2 respired by the peas. Because you take the number of drops it took to clear your standard solution, right? Say it was 177. But then say you had your pea solution and the peas had been in it for a while respiring, gradually turning it closer to clear. And then you take your peas out and get rid of them. And then you just put your drops in there and say it took 120 drops to clear that solution. Well, it took 120 drops to clear it. So we know the peas didn't do that part. So we subtract that 120 from your 177, and that net number of drops is how much clearing the peas did. Because that's the equivalent to how much they did in there. So we got the set in the blank. What's the name of the enzyme we tested in lab? I've covered that up there. That was peroxidase. I don't know if it'll ask you what it was from, but it was from a turnip. The substrate that was converted to water was the hydrogen peroxide. The reagent that turned brown when oxidized was the guaiacol. And I have, because a lot of these are down here in the multiple choice further down. What reagent did we use to test for starch? That was iodine. And remember, when it's negative, it's going to be yellow like the iodine, and when it's positive, it turns black. And in the enzyme lab, I've covered that too. If it's a dark color, it's higher enzyme activity, higher absorbance, and lower transmission. If it's a lighter color, it's lower activity, lower absorbance, and higher transmission. So if you remember that, you'll be able to answer questions similar to this no matter how they're worded if you just remember the basics there. Because you could be asked about their absorbance value, their transmittance value, or what their activity was. Okay? Number 9. The substrate that was converted to water was the hydrogen peroxide, which is H2O2. So
What
we got
was the reagent that tested for protein?
tested for protein, that's Bradford's reagent. And of course that turns bright blue when it's positive. And it's just like that burgundy color like it was out of the bottle if it wasn't. So which one showed the most enzyme activity? Cuvette 1 or 2 in this. It'd be 1 because it was darker. The optimal pH or temperature for peroxidase activity. The optimal pH is 5. And the reason it was 5 is because since you're doing a redox reaction, having extra hydrogen ions available to shuttle electrons, basically, because you've got gain and loss of electrons, the hydrogen ions help facilitate that. That's why the pH 5 was optimal. The temperature that was optimal was, of course, like the temperature for life, about 30 degrees, 40. And then what organism we used in lab to measure the aerobic cellular respiration? That was the peas, of course. Now it's the paper chromatography ones. These are the questions from the lab that we just did, right? You have your solvent front, which is 10 centimeters, and your pigments each travel a different amount. And it's not even labeling what they are here, it's just labeled them A through E. So you write your answer, you just want you to calculate the RF value. So when you're calculating that RF value, RF value is equal the pigment distance divided by the solvent front. So, it wants to know the RF value for pigment D. Pigment D traveled, I'm gonna do it up here. It traveled 7.5 centimeters. And you wanna divide that by your solvent front, which was 10 centimeters. So its RF value would be 0.75. And then your highest RF value would be 1, which would be, in our case, and these are all in centimeters. Oh, all the RF values are not centimeters, though, it's just a value. In our case, in our lab, our beta carotene was right there in the solvent front, wasn't it? It was wet right there at the line. No, yours worked, didn't it? In your lab? Did your pigments separate okay?
Yes.
Okay, and it was wet right there along the beta carotene line, wasn't it, when you first pulled it out of the jar? Right, so the RF value for our beta carotene would have been, I'm just gonna call it BC, it would have been 10 divided by 10, which is 1. That would be your highest RF value you can have, because that would be the pigment that's most soluble that's traveling with the solvent. Right. But when you get questions like this, they'll all be below 1 in your RF values. So in this case, the RF value for pigment A would be 0.25, because they're all divided by 10. And we try to do this on exams too. We try to set up most problems, you do them in your head. You don't need a calculator, because divided by 10, you're just moving your decimal in one place. So yeah, and that's showing your work too. So you need to understand, when you show your work, you don't necessarily have to write this, but it helps if you remember that, to write it down. It's like, then you won't get it wrong. But just be sure you write this. That's showing your work. You need to show that you understand how you calculated that RF value. So based on the data above, is pigment D more soluble or less soluble than pigment B? Alright, so we'll go over this too. I've got this little mess already, don't I? I need another chalkboard. I could really cover 4 labs easy if I had 2 walls of chalkboards. Alright. I'm going to go ahead and draw a line here. So, the farther it travels, equals more soluble. So, if pigment D traveled 7.5 centimeters and pigment B traveled 4.5 centimeters, then pigment D is more soluble. I'll let you guys write notes to that effect. So based on the results above, does pigment D have more polar groups or fewer polar groups? It would have fewer polar groups. The farther it travels, implies that it has fewer polar groups, because the more polar groups it has, the less distance it will travel, because it's associating with your fiber of your paper. What's it called? Cellulose. So the farther it travels, it's more soluble, less polar. According to the lab results, chlorophyll A absorbed the maximum amount of sunlight at what wavelength? So when you were taking the readings, you guys really should bring your homework stuff with you for reviews. That way you can just look at answers. Huh? Huh? the actual lab information. But you need to know, it's, I thought I had that before, synthesis folder too. But I think I got rid of a lot of stuff when I did this. There we go. So basically, that's a chart of them. So what wavelength did chlorophyll A do max absorbance at? That's your wavelengths of light down at the bottom.
420.
Yep, 420. And yeah, I was saying, sure people could make jokes about that. But 420 is the highest absorbance, and what does that mean as far as its oxygen production at that wavelength? So, I need more chalkboard. I'll go erase all this. So right here I'm gonna write photosynthesis. So. Higher absorbance equals, I'm just gonna put under here, oxygen production because more photosynthesis. That's what your higher absorbance means. That's where it's doing photosynthesis. Now your lower absorbance could almost say lowest. Let's say lowest absorbance. Goes down there at the bottom. No photosynthesis. And here's a surprise for you. That's what color the pigment is. Because if it's not absorbing the light, it's reflecting it. So you see the light that's reflected, and that's the color you perceive. And this was the biggest, the more I've taught this, it's become almost like an existential crisis-like thing, because it makes me realize that colors aren't real. Nothing is actually deep color you see it at, because what you're seeing is just the wavelengths of light it reflects. And I'm like, wow. So, because without light reflecting, nothing has any color. So the lower wavelengths is where the light's reflected, and that's what color you see. You will be asked a question like that on the exam. You'll get maybe a graphic data or something. It'll ask you, at this wavelength, is photosynthesis occurring or O2 production occurring? And then it's going to ask you what color that pigment is. And you'll have to be able to tell that from the graph. And from the graph, you can tell chlorophyll A is green because it's not absorbing any light in the green spectrum. The carotenoids are red-orange. It's kind of weird because they're flat down this. Oh yeah, because there's no, yeah, the carotenoids, that's the color they are is down here because they're absorbing light down at that end of the spectrum. So the carotenoids are doing the absorbance in the purple-blue range, but they appear yellow-orange. And then, of course, the chlorophyll B is in different wavelengths also. And that's why, I'm not sure if that explains it really well, but it seemed to me The difference I perceived on the paper was that the chlorophyll B was like a darker gray green, and the chlorophyll A was a brighter, like leaf green, yellow green color. So I don't think the graph really shows that. And then, okay, so would you expect oxygen production from a plant if you exposed to the light at 540 nanometers? Would you expect photosynthesis? No. Because that is reflecting all the light back and not absorbing any. And that's basically in a nutshell the stuff we've done. Now, you have the option of practicing blanking a spec, changing the wavelength, setting the blank, go through your steps, make sure you memorized it because you'll be graded on all the steps of that. Whereas most stuff with just 1 or 2 points for like the scale and balance, I think there's like 4 points of stuff you do with the spectrophotometer, so it's worth your points, get it right. And then also, you're welcome to get out a microscope and look at your Elodie leaves. You have to know how to focus the microscope yourself. You have to know how to, once you get it focused, make sure you've got it bright enough and that you've got the diaphragm closed enough to see the chloroplast clearly and the organelles. Because that's a point if you don't use the diaphragm right. So there's points with all of it. Bringing it up to focus, it's really hard to score people that can't even find it. And then you have to get it in the focus group and then see if they do the best. And so it helps to know all of those steps. And you're welcome, like I said, Lily and I will help get stuff out, you know, if you need slides and stuff for the microscopes. Just go ahead and get your microscopes when you do it. And if you have any more questions, ask me right now. So, do you feel like I covered a lot?